Welcome to my channel Adhyan with Flow. Today we are going to discuss about structure of protein. Okay, now we know that protein is the structural and functional unit of any kind of enzymatic activity, any kind of hormonal activity, any kind of signaling activity. We already discussed. Wait guys, I will show you. I already give you brief introduction about protein. Some functions of proteins. Look at there guys. They work as a ECM, example is collagen. They work as an enzyme, example is trypsin. They also work as a hormone, example is insulin. They work as an immune system. They form antibodies. They work as a receptor also. They work as a channel, carrier, pump also. They can transport molecule outside to inside the plasma membrane and inside to outside the plasma membrane. These are channel. According to their name, GLUT, they transport glucose into the cell. Means this is the cell. And here channels is located, and with the help of glucose come inside the cell. I already described their function, how they form. We all know that what is amino acid. That amino acid contain one side carboxylic group and another side amino group. We already discussed 20 types of amino acid with their special properties and their structure also. We already discussed. I'm telling you guys where I teach all the things. Wait guys. In amino acid we discuss here guys. I completely describe in elaborate manner amino acid, their type, their structure, their mechanism. I already described lipids, nucleic acid, everything in the starting of the chapter. So you can watch first this video. Okay, now let's start. In structure of protein, according to their structure, protein divided into 4 parts. Primary structure, secondary structure, tertiary structure, quaternary structure. What is the primary structure? This amino acid bind with another amino acid. This amino acid bind with another amino acid. This amino acid bind with another amino acid. This is the linear chain guys. Look at here. This is the linear chain. I'm improving my writing guys. I'm trying to improve my writing. So the linear orientation of protein chain, linear orientation of amino acid chain, we call it as a primary, okay, not like this, primary protein structure, isn't it? And now if someone asks what is the structure of amino acid, I know I explain all the things, but again they contain amino group, they contain carboxylic group, NCC rule guys, remember NCC rule, and here is any kind of R group. It may be carbon chain, and it may be ring like a structure like we study now wife. I give you code guys. Okay, they contain one side amino acid and another side. Okay, now how pairing going to occur? This is the one amino acid and another amino acid. How they can pair? I'm writing here, I'm making here NCC, again they pair NCC, again they pair NCC, again they pair NCC. Those students who already watched my NCC rule, they can easily understand that this is the one amino acid, this is the other, third amino acid, fourth amino acid. This is the amino group, this is the carboxylic group, and this is the carbon. Look at here, amino group, carboxylic group, and center carbon. Here hydrogen attached, here R group is attached. So this is the sequence. We call it as a primary structure of protein. How many amino acid present here? 1, 2, 3, 4, 5, 6. Now notice here is a bonding between two amino acid. Be focused, okay, be focused. This is not easy, okay, this is not a halwa. Notice one amino acid connected with another amino acid with this bond. We call it as a peptide bond. This is the covalent bond guys. Actually covalent bond means they share their electron, okay. Covalent means strongest bonding. Yes guys. So what you understand with primary structure, what you understand guys, whatever I'm explaining here na, already written here, okay. Now primary structure, what you explain, one amino acid bind with another amino acid with the help of peptide bond. With the help of peptide bond, I'm zooming this bonding. I'm gonna zooming this bonding. Wait, little bit. Okay, here is another amino acid, H3 and CC. Here, what happened with here? I'm zooming again this structure, NCC OH group. And now what happened this? H and HCC. So this is the one amino acid, clear guys? This is the another amino acid, clear guys? And here removal of water. So due to removal of water, this bonding going to occur. Now clear my bachcho. After removing this bind with this nitrogen. Okay, so these kind of bonding is primary structure form. Look at here guys. Look at here. Amino acids, they are sequentially linearly arranged. They make a linear chain. They contain peptide bond. The special feature is primary structure contain peptide bond. Now let's move towards secondary structure. Look at here guys. Secondary structure divided into two parts, alpha helix form and beta plated sheet. Guys, we know that this is the primary structure, but this is not a functional structure of protein. No, this linear structure of protein does not take participate in the functioning of the body. What they do now, after peptide bond, now they form hydrogen bonding. Look at me guys, I'm explaining how guys. Listen to me guys, what happened with this chain, I'm explaining here. Okay, we know that this is the linear structure, but this linear structure not functionally active. They have to be folded in this manner. They arrange like N C C N C C N C C N C C. Now what they do? They present in their folding structure, and now what they do? They make hydrogen bonding. This is the special feature of primary. They have peptide bond. Secondary structure of protein, what kind of a special feature they have? They have peptide bonding, and also they have hydrogen bonding guys. Yes. Okay, what they do? We know they can bind with another amino acid with hydrogen bonding. They can pair with another. Look at guys, they make hydrogen bonding. These are the hydrogen bond, and here is the bonding is peptide. Due to this bonding, means this amino acid pair with this amino acid by hydrogen bonding, then what happen? This amino acid and this amino acid come closer. So now assume this is the first amino acid and this bond with this amino acid, then they close. If they bond with this, then they close, so they oriented in this manner. Now, what is beta plated sheet? This is the helical structure, like this, and here is hydrogen bonding between them. Okay, now this structure also contain hydrogen bonding as well as peptide bonding, but the difference is they arrange like a sheet, this, like this manner. They make a orientation, perfect orientation. Okay, so this is the difference between alpha helix and beta plated sheet. Alpha helix is more active and functional form, they are very static. Now let's move towards the tertiary. We know that enzymes they make their active sites, so they can substrate with bind and make product. So active site formed by tertiary structure of protein, they recoil itself, they contain peptide bonding, they make hydrogen bonding, and they also make disulfide bonding. They make hydrogen bonding, they make sulfide bonding, they also make peptide bonding, and the supercoil. This is more active form. This is more active form, guys. This is more. They overlap. What is the special feature of this tertiary structure? The R group come outside. The R group come outside, and here in a center part, this is the hydrophobic region. We know now amino acid contain one R group, so R group come out from the surface. This is also a functionally active form. Now let's move towards the quaternary structure. One of the best example is hemoglobin. They made up with 2 alpha subunit and 2 beta subunit, like this. Here is multiple polypeptide form and make 1 alpha subunit. Here multiple peptide chain fold and make another alpha helix, alpha. Now this term to beta, and they make supercoiled, chain is supercoiled, and they make beta subunit, and chain is also supercoiled, and they make what kind of bonding present? Complete bonding, guys. They contain peptide bonding, hydrogen bonding, guys, hydrophobic bonding, disulfide bonding, both kind of bonding. If you want to study in depth these kind of bonding, you can watch my video bonding, chemical bondings. You can watch my video chemical bonding. I explain there multiple bondings. So now this is the quaternary structure. Okay, guys, so this is the structure of protein. So let's start reading one by one. What is the primary structure? Okay, they contain amino group and one side contain the carboxylic group. One amino acid bind with another amino acid with the help of peptide bond. This is for primary. Look at here, the first amino acid is also called as N, because here is N terminal and here is C terminal. The last amino acid is called as C terminal. This is chain, guys. This is the chain. Now last is called C terminal, and here they called N terminal. A protein thread does not exist. Okay, look at here. Does not exist throughout as extended rigid rod. They are not exist in this form. They have to coil. They make a helix. Of course, only some portion of the protein thread are arranged in the form of a helix. In protein, only right-handed helix are observed. We call it as a secondary. They direct told me. They don't say any kind of peptide bond. They don't tell about hydrogen bonding. Okay, guys, but I explain everything. In addition, the long protein chain is also folded upon itself like a hollow wool ball, giving rise to tertiary structure. This gives a three-dimensional view of the proteins. Tertiary structure is absolutely necessary for many biological activity of protein, like enzyme. This is the enzymatic form. They make active site of enzyme. Active site. Here substrate can bind. Some proteins are assembly of more than one polypeptide. This is the one polypeptide chain, this is the another polypeptide chain, this is the another multiple polypeptide chain. The manner in which these individual folded polypeptide or subunits, this is the two subunits of alpha helix and this is two subunits of beta helix, are arranged with the respect of each other. A string of sphere arranged one upon each other in the form of a cube or plate is the architecture of a protein, otherwise call it as a quaternary structure. I think I explain everything. Okay, so this is all about protein structure. Okay guys, this is the bonding here. Okay guys, bonding, I already discussed chemical bonding in my previous video, but I again told you, I again explain you, but in my next video guys. So let's meet in our next video. So that's all for now, and guys, if you still not subscribe my channel, subscribe here. Thank you. Keep smiling. Thank you.